right, let's talk Ediacaran creatures because they're pretty cool. We're going to keep our little ctenophore up there since ctenophores, if they're that diverse at the bottom of the Cambrian, probably date back to the late Ediacaran, which is when we actually have that ctenophore like embryo. So, yeah, probably. Here are some, we'll start with paleoecology. So, the easiest thing to do with paleoecology is say, okay, what fossils do we have? Now, in the Ediacara Hills, you'll remember this is death mask style preservation for the most part, although sometimes it's layered up, multiple layers of death masks, um, like in the one fossil that we have from there. And you'll see that there are a bunch of these with this brown stuff in there. That is Aspidella terranovica, I believe. And Aspidella is a weird one because it kind of looks like it's there with the animals, but it also just looks like sort of a blob that's kind of halfway buried, and you can't see any parts on it, but that's in part due to death mask preservation. Some of them are really big, but usually, like Cyclomedusa gets quite big, but Aspidella tends to be fairly small. There's one group that says that this may just be little tunnels that are made by bacteria under the ground making methane, and so they're putting methane up, and then the methane is going to find its way up and sort of carve out a little tunnel, and then when it gets to the surface you'll have bubbling, and so you have like a little crater that's bubbling all the time from all the bacteria, and so a whole big area of methane producing bacteria would be sort of funneling it up like a little chimney, which is just like the path of least resistance that happened to be made first, and then gets widened out over time. Is that what they are? They've also been proposed to be holdfasts for things called rain geomorphs, which are like a little sea frond that may be laid down and then stood back up. But we'll get to all that. What you can see here, though, just sort of zooming out, is that wow, at most sites, they are dominated by one organism. Like, look at this yellow organism. And it's what? Yeah, 90%, 95% of what you find. And then there are a few other things there. If you're thinking in terms of ecosystem, these creatures were at least originally thought to be all eating the same thing, which is the microbial mat that's on the ground, which is a mix of algae and bacteria. It's just sort of a thick, snotty looking layer, basically a big biofilm. And yeah, the idea is that they're all sort of grazing on that. But if you were on the Serengeti and you say, oh, there's this many of this thing, and, like one of those, that's like the ratio of gazelles to cheetahs. Yeah, there's a lot of gazelles, not very many cheetahs. So we potentially do have predator-prey relationships. Do you always find the light blue thing where you find the yellow things? No, but when you do find them, there's very few of the light blue ones and a lot of the yellow ones. So the light blue here is Dickinsonia, which is one of our favorite organisms for sure. Yeah, so interesting, I think. Yeah. All right. What you get in Ediacaran developmental biology is something that's unusual in the sense that it is very rare in higher organisms. There are some types of jellyfish. The name is escaping me. It starts with an S. And they'll have this as well. It's called glide symmetry, where instead of being bilaterally symmetric and having a thing off each side, you're going to have glide symmetry, where you have one here, then one here, then one here, then one here. So every new element is added sort of up and to the side from the old one, like at a right angle. And that's what you get here. We see this in plants, pretty common in plants, but not common in animals. But all the things that we would call plants, that we would think of as multicellular plants, do not show up until fairly late. I think the Ordovician is thought to be the time when they first appear. Either Ordovician or Silurian, actually. So, come back. There we go. Yeah. Yeah, so in an animal, that's strange. But here is apparently an animal These are rangiomorphs. And here are different ones. You can see that they have glide symmetry. They look like fronds usually. They stand up as if they were like a little fern. Off the ground, they'll have a holdfast that appears to anchor them to the floor. And yeah, there's just a wide variety of these things. Sometimes the holdfast is really little. Sometimes it's basically a great big holdfast with glide symmetry on top of it. Yeah, that's just how they are. Here is another. These are Ernietta, or things related to Ernietta. Ernietomorph. So rangiomorph, things that look like rangia. Ernietomorphs, things that look like Ernietta. And they look like little tongues, but with glide symmetry. And they you find them all sort of piled up like this in Namibia. Don't know if they are sort of all eating a big blob of the same sort of material or what. Yeah, it's hard to say. Oh, here we go. One moment. We lost power. That's why we keep losing an image. There we go. We got power back. All right. All right. Here are Dickinsoniomorphs. So we had rangiomorphs. Rangea is the thing they're named after. We had Ernietomorphs. Ernietta, Ernietta. And we have Dickinsoniomorphs, named after Mr. Dickinson. Dickinsonia is the organism that these are named after, and there are several other ones, and you don't have to learn the other ones. Yeah, unless you're going to study this stuff. They're pretty awesome. You know, you can learn them if you want to, but I don't think it's necessary for anything like a test. Yeah, learn it for fun. So here's Andiva, here's some Dickinsonia, here's a reconstruction of a Dickinsonia. You can see they've reconstructed it without glide symmetry, even though it probably does have glide symmetry. But this is the point when this one was made where people were starting to doubt that, or maybe this is a group that, you know, professionally doubts it. They're like, maybe this is a bilaterally symmetric organism. It's probably not. Yeah, it's just another Ediacaran glide symmetric weirdo. All right. Here's Charnia masoni. This is a rangiomorph. And so they're just looking a little bit closer, and you can see that, oh, okay, it's got these big fronds. And on each little frond, it's made up of smaller units that are almost at a right angle to the rest of the frond. You say, oh, okay, so it's organized in big ovals that are made of smaller perpendicular ovals. And then if you look closer, those smaller perpendicular ovals are actually made of smaller ovals that go perpendicular to the perpendicular. So little tiny ones like that. And they haven't been able to see this, but the idea is that probably those are made of itsy bitsy ones that are just like little boop, 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 perpendicular units. So it's a very simple sort of organization that if this was a computer program could be carried out in basically 2 steps where you start at a base and then you add a branch to the left and then you move up and you add a branch to the right and just repeat that process and you will generate an organism that looks like this. It's a fractal sort of organization, you know, like those really cool patterns with repeating units that are made of the same mathematical construct. Yeah, and basically fractals are what these look like. So here is the simple fractal interpretation of it, and here's the actual fossil. And yeah, it's kind of a spot the difference. There's not a lot. And so all they're changing here is how far up you go, how wide each of these units is, which are just like constants you put into this equation. And honestly, yeah, it does a pretty good job of reconstructing these organisms. So there you go, little rangiomorphs. Here's another weirdo that I like a lot. It's called Fractophusis. Fractophusis is right there. And these guys are all found at Mistaken Point. It's showing some other organisms here. Here's a, see the holdfast and the stem and the frond right there. And that's a Charnodiscus, which is like Charnia, but with a big fat base on it. Here's our Fractophusis right there. Here's an Ives hedia. They are called pizza blobs. We do not know what those are. They just are chaos in a little blob that was standing up above the bottom. And yeah, good luck trying to figure it out. Here's an interpretation from this 2017 paper, and Duncan McElroy is pretty good at this. I don't know Suzanne Dufour, but it's a good paper, so maybe I should. Yeah, maybe that's on me. Here is the idea that you would have an oxygen gathering surface. You would have hydrogen sulfide and methane, which we think were definitely coming up from bacteria from the ground, or that are potentially being metabolized by commensals, or potentially, I guess, even the organism itself. So you'd have these reactions happening with the little endosymbionts, bacteria, being part of that. And it would puff up to get oxygen in there. And then once the oxygen was in, it would close back down and do the reaction. So it's like it would open up, let in some air, and then oxidize those materials that came in, and then puff up again, maybe expel waste and fill back up with oxygen. Kind of like breathing. Huh? Yeah. Yeah. So yeah, puffing up to pick up oxygen and pulling it down to go send it around to do chemical reactions that produce energy. That sounds a whole lot like breathing, actually. Yeah. And so the idea there is that these organisms, these Charnia, all of these things appear to be hollow, and the hollows would be there because these are reaction chambers. So it's like the whole thing is both a lung and a digestive tract. And the glide symmetry is just an easy way to multiply these, but still have them linked together so that you can increase the overall amount of energy that's made inside the organism in the sort of biologically cheapest way. It's just like an efficiency thing. You're just increasing the size of your factory so you can double output, something like that. All right, so here's a Fractophusis, and this is death mask preservation. So this is an inverted, like a little hole in the ground that was made, or yeah, this is a hole in the layer that was over top of it, like that. There we go. And what you see with Fractophusis is here's a big one, and then here's one that is much smaller, and it's about the same size as this one, which is much smaller. And if you zoom out, you actually see that there are huge ones that are surrounded by large ones. And it'll usually be 4 of those. And then the large ones are going to be surrounded by 4 to 8 small ones. And the 4 to 8 small ones are going to be surrounded by 4 to 8 little tiny ones. The idea being that these things grow and they branch out and they set out a little projection from themselves, and that becomes the children. You can just barely see here. There are little traces in the rock that appear to connect, like an umbilical cord, the large one to the medium ones to the small ones to the tiny ones, and the huge ones to the large ones. Pardon. So it may be that every year these things would basically divide, put out little roots, and grow a new organism at the end of that. That's something that grasses do. It's called stolon replication, Stolon. Still kind of hypothetical for Fractophusis, but I haven't seen any papers that come up with a better explanation for this, and it's kind of cool, yeah. So yeah, there's a little landscape dominated by Fractophusises, fusi. Here's another thing about these rangiomorphs. So this is one, and it's showing that this yellow here is the fossil of the actual animal. This is much deeper, more three-dimensional fossil. But right next to it, you get what appear to be little ghostly images of the same creature, which could be where it is basically laying down at night doing chemosynthesis and then puffing up, but instead of just puffing up, it's puffing up and standing up in the morning, which would be much more efficient. You'd get higher in the water column, you could collect more oxygen the closer you are to the surface. And so potentially that's an evolutionary adaptation for this Ediacaran type of life. Yeah. And yeah, they do see that at, I believe it's just at Mistaken Point. But you get little patterns of the frond right next to imprints that look like they are also of the frond. And the different positions may just reflect differences in the current, where the tide is coming that day, something like that. Or yeah, I don't know, there may be some other control there. Here is Dickinsonia, and so here's a Dickinsonia that they're drawing once again with bilateral rather than glide symmetry, which could be true, but I suspect it's probably glide symmetry. I'm going to lean that way. But yeah, you've got each of these units has a little rounded tip, so like a little bulge that comes out the sides, and there are impressions where it looks as though the whole thing can maybe contract and expand the entire body. So here's a contraction impression, like it was over here, and then it's either moved itself to the side or expanded and then contracted. Yeah, and you get different sizes of units. So you'll have an area at the anterior, and that anterior sort of main unit is going to be maybe the first thing that's put in along with the posterior, and then you'll have branching next to the posterior. So these will be the oldest parts, these will be the youngest parts, and it'll just keep erupting from the bottom and pushing the other ones up. And so this is an organism that can basically undergo constant metamorphosis as each new layer is put in here and then sort of squishes the old layers together and changes their shape. But if it's a hollow creature, that you can imagine how that can happen. Yeah. Here are Dickinsonias of different ages, from the tiniest little baby, look how cute that is, the giant little anterior unit, to quite big ones over here. We can see it's just a little tiny unit, and they've got them organized so the anterior unit is on this side of all of them. Yeah, there they are. So, when studying very weird creatures like this, which frankly, from the early reports, could be plant, fungus, animal, or some mix of those things, or another thing that's none of those things, yeah, this is an area of hot debate. It's often good to have a look at how they develop and see if that gives you any clues. And so here is the ratio of total width to total length, and so they're proportionate. So it gets wider as it gets longer. But each of those little segments starts out really short. So that means each segment would also get longer and wider, but probably thinner over time as it gets pushed up into the toward the middle of the body of the animal. Here's the number of modules, and you can see the number of modules goes up kind of proportionately at first, and then it kind of levels off and sort of stops. So there seems to be a maximum number of units that we're at least going toward. It would probably, you could imagine this equation leveling out like that, which would be the maximum size. And maybe that's the maximum sort of compression you can do on the thinnest units around the middle, and have them still work, have them still serve whatever function they're supposed to be serving, because that's up in the air. Module width related to module number, normalized for if there's like a break in the fossil, and you can just say, well, the other side's probably the same even though it's not here, and you gotta do some estimating. You'll see that module number is proportionate to width in early development, And then in large individuals, they sort of drop off and then they go down, 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 like that. So the modules get smaller as the organism gets bigger. So it's not that the modules are just growing indefinitely, they are actually getting much smaller. So it's kind of like division, or that there's a certain amount of space or a certain amount of distance you can be from the center before transport of nutrients or whatever it is they're doing is no longer efficient, maybe. Here we're showing the path of these. So here's a very early little segment being formed, if segment is the right word for them. It's the word that they use. Why do they have an A in segment? Let's put that as an E. There we go. Yeah. And here is that same one that's pushed up toward the top. You can see the difference, or the distance between red and blue is going to be the same. I think it's like 7 in both cases. And then here is the new segment that was put in down here. It's green. And so they start out like this and very short, and then they get longer and flatter, and then they'll eventually start to curve the other way and get super wide like this, and super skinny. And that's how the organism is put together. Now, this paper is pointing out that if you just take the features that we can see here and try to put those on the tree of life, there's a temptation to say, well, this is probably a sponge, this is probably an arthropod, and maybe this is too. But I think what this paper is saying is maybe don't do that. Think of these as a crown group from which all animals come. And in that sense, let's look at these. Because if you take a little fly embryo, the first parts that develop are going to be the head and the tail. And then we're going to put in segments in order, going from this way down to that way. And so we'll just keep branching near the tail, so the posterior end. And the anterior is just going to stay as it is. And so here we have the two oldest parts. And here are the newest segments that have come in based on comparing lots of these at different stages of growth and assuming that segments start small and then get bigger, which I think makes a lot of sort of sense. And in that case, you would say, oh, well, this probably isn't the anterior. This is probably equivalent to the posterior if they are using similar genes to produce this as modern animals. And if you have a sort of a head and a tail that develop and then remain sort of static while other units are added around them in a particular order, yeah, there's a similarity there. And so, yeah, you have growth down here and growth up at the tip of these fronds. So the fronds are a little weird in that sense. But, yeah, I don't know. What do you think of that? So the idea is that you could place rangiomorphs down here. Like, they have some features in that they are hollow, potentially filtering water, potentially grabbing air and doing chemosynthesis with bacterial symbionts. And that could be similar to porifera as well as ancestral to these other animals. Or you could put them up here and say, no, these are maybe past where I would consider sponge-type morphology, but these may be the step that actually is ancestral to all the other types of morphology. And here they're just saying that, I don't know where ctenophores go. This was right after the ctenophore paper that kind of rocked the world, the first genome. And so they're being very cautious, which is the right thing to do. And probably the ctenophores are the earlier branch before porifera, based on current understanding. But then, yeah, a Dickinsonia, minus some of its organization, or a Dickinsonia morph like Andiva, is a placozoan. Basically, those are the little flat things that swim through the water in no particular way and have digestive patches on the outside of them. Or you could say that, you know what, all these guys, they're starting to develop bilateral symmetry. So they're kind of on the way to that. And this glide symmetry, if we just alter the equation instead of going up, over, and perpendicular, and we make it in a slightly different direction or right beside the other one, or we do two at the same time, we just double those genes, then double glide symmetry looks like bilateral symmetry. and you could have that underlying hydrozoa and anthozoa, which are two different cnidarians, and all the bilaterally symmetric organisms, potentially. Yeah. So that's the point of this one. It says maybe we should be thinking of Ediacaran creatures as if they are in a crown group, not as if they are members of any particular group. It might be wrong to try to look for modern affinities. But what we see over and over in the fossil record is that there is a reuse and then a mix of loss and duplication of genes. So we don't really get more complex. We make more copies of the things that we retain, and we lose things in different branches. And so basically, evolution is the process of paring down these early, maybe pluripotent, like things that could have gone a lot of different ways, sets of genes and organisms, and just partitioning those. Like, and you could imagine the environment playing a pretty big role in that. Because if there is an unclaimed food source of some sort, something that could be eaten, then why wouldn't one of these creatures eat it if it can? And if it can consume that food source and utilize it without needing certain parts, then eventually, over time, those parts are going to get mutated, but there's going to be no penalty. And if you get a genome deletion that loses some of those parts, it's actually faster to replicate a shorter chromosome. And so you might be able to increase or rather decrease the amount of time between generations. You might be able to increase overall fecundity, like how many children you have, basically, if these can be called adults or children. I don't know. Yeah. So yeah, think of evolution in that way, I would say. And that'll be pretty useful. All right, review questions. Let's take a look at these. Let's see what the heck we put in there. What characteristics are shared by many Ediacaran organisms? Glide symmetry for sure. Fractal organization for sure. How are those different from what we see in later organisms? You get both of those in plants, but they've been pretty much lost in animals. Siphonophores is the word I was looking for earlier. And it's not really glide symmetry, but it kind of looks like it. And so you get it in the tails of some of these long, crazy looking jellyfish. There is, or at least was, a very nice siphonophore focused Twitter feed. I'm not going to say the new name of it because it's ridiculous. And it would just post pictures and videos of these amazing creatures as they were tending to them and feeding them and studying them in the deep. Yeah, so pretty awesome. Describe the development of a Charnia frond. So Charnias are growing a little bit at the stem, and they're growing at the tip where the smallest units are. And so those are the newest parts. And then the oldest parts are probably the holdfast, at least the center of it, and the lowest, largest units coming off the side. Dickinsonia and Andiva, these are both Dickinsonia morphs. Charnia is a rangiomorph. So maybe that's what I should have put there, of a rangiomorph or a Dickinsonia morph. That would be a lot more sensible, wouldn't it? Yeah. So those are going to be fixed at both ends, and they're going to insert new segments here that then are going to move up as new segments are inserted below them. They keep pushing them up. And the organism will get both wider and longer up to a certain point, after which it kind of stops, like the number of segments is kind of limited, and you just get overall growth of the creature. So what's a fractophusis? Nobody knows, but it's a fractal symmetry. It's sort of stuck to the ground, and there are little chains linking it to a number of equal sized, and presumably size increases with age. It may not be linear because you may get to the winter and things may not grow as much. That's what you get in modern corals and stuff. You know, it's temperature dependent. But yeah, imagine it as just like steady growth. And so the bigger you are, the older you are. But yeah, maybe stolon replication. Yeah, that'd be pretty cool. What are the characteristics of Ediacara type preservation? Death mask. How did they form? Sand that turns to sandstone. It makes a death mask. What do they preserve? the shapes of the outside, even of soft-bodied things. What's lost? Well, they tend to compress the thing because if the thing was actually soft-bodied and squishy, and you put a great big, bag of sand on top of it, it'd get kind of flattened out. Yeah, so they're probably a little bit flattened. Usually the organic components are lost. Um, What's the evidence for Ediacaran organism motility? Well, you've got the Charnia fronds, or Rangiomorph fronds, that are down and have a bunch of other little positions next to them. You have evidence of possible contraction in Dickinsonia, where it looks as though there's a rim of what it was covering, where it's sort of eaten into the biofilm, and then it's retracted back over here. You also have feeding traces of Dickinsonia and a really cool organism called Kimberella that had something like a rake and we think was related to, like on the way to being a primitive mollusk. Yeah, those are the main things. How did Dickinsonia move? Good question. But there's some evidence for contraction, and there's some evidence for Dickinsonia-sized holes in the biofilm being arranged maybe 6 inches or a foot apart, and so it's like it sort of picked up, moved over, and then digested everything under it, and then picked up, move over, and digested. That's the current understanding. How did rangiomorphs move? Inflation and deflation. Inflation, deflation. How did Kimberella move? We didn't even talk about Kimberella much. We will later, but this seems to move sort of backwards and be scraping along behind it. So it'll back up and it'll scrape all the biofilm into its mouth and then back up and do it all again. And so you get these trails of little scratchy marks where they've sort of mined up all the nice biofilm and eaten it. And yeah, the creature's constantly moving away from what it's just eaten. So it basically sits on its food and then it eats its food. Yeah, that's how I would describe it. All right, that's more than you ever needed to know about Ediacaran organisms, but thanks for sticking around.